EdvoTech tips, ingredients for PCR success. Hi, my name is Dr. Danielle Snowflack and I am a scientist at Edvotek. Today, we're going to discuss a very common biotechnology technique, the polymerase chain reaction, or PCR. This technique lets researchers create billions of copies of a specific region of DNA in vitro. PCR is a very common technique in the laboratory. It's not a perfect analogy, but we can think of PCR like a DNA photocopier. You choose a specific page you want to copy, and if you have enough paper, you can reproduce it over and over again. But how does it work? And what do each of the components do? I like to think about PCR in terms of DNA replication since it follows the same steps. First, we have to separate the two DNA strands. In a cell, the enzyme helicase unwinds the DNA into single strands. In PCR, we separate the strands by heating the sample to near boiling. This breaks the hydrogen bonds between the DNA base pairs. This is called denaturation. Next, if we were in the cell, the enzyme primase would build short RNA primers on the DNA strands. This allows DNA polymerase, the enzyme that builds DNA, to form bonds between the nucleotides. DNA polymerase can't build DNA on its own though, so it needs these primers to get started. For PCR, we designed short synthetic pieces of DNA that target a specific DNA sequence. These are our primers. We adjust the temperature of our sample to something between 40 and 65 degrees Celsius, which causes the primers to bind with its complementary base pairs. This temperature is based on the thermodynamic properties of nucleotide binding. We call this annealing. Once the primers are bound, DNA polymerase can start building DNA using the existing strands as a template. This is extension. In replication, we copy each chromosome in an organism. In PCR, we raise the sample temperature to 72 degrees Celsius, which allows polymerase to attach to the open ends of the primers and fill in nucleotides, thus creating copies of a section of the original DNA molecule. Now in replication, we're done until the cell divides again. For PCR, we're going to repeat these steps 25 to 35 times to create a ton of DNA. Each cycle doubles the number of copies in the tube, giving us many millions of copies of DNA before the process is over. To amplify DNA using PCR, we mix primers, template, and DNA polymerase together in a tube and then place them into the thermal cycler. So, what role do each of these reagents play in PCR? So, as I mentioned before, primers target our sequence of interest. They're designed to flank the DNA sequence we are hoping to amplify. Our template is extracted DNA that contains the sequence that we want to amplify using PCR. This can be any kind of DNA, plant, bacteria, or even human DNA that your students extract from their cheek cells. DNA polymerase is the enzyme that builds DNA from nucleotides, using the template for reference. Early PCR experiments used DNA polymerase 1 to amplify DNA, but there was a problem. Since DNA polymerase is a protein, it was inactivated every time the sample was heated to boiling. The discovery of the heat-tolerant Taq polymerase revolutionized PCR because you didn't need to add new polymerase at every extension step. Our Taq polymerase is provided as a shelf-stable PCR bead, which includes the enzyme and nucleotides which we need to build DNA. The beads contain buffer and salts as well to make sure that the reaction is optimal for the enzyme to work. So, what happens if we leave one of these components out? Let's try it. The kit I'm using here is EdvoTech 371 DNA fingerprinting using PCR. The kit includes primer, PCR beads, and several samples of DNA template, each which produces an amplicon of different sizes. I'm going to use DNA sample 1 in this experiment. I'm going to set up several PCR samples for analysis and put them in our EdvoCycler Junior. Our first sample contains all of our reagents, primer, template, and Taq plus nucleotides that are in our PCR bead. Our second sample replaces primer with water. Our third sample replaces the template with water. And our fourth sample leaves out the PCR bead. So what do you think is going to happen in each sample? Using the kit instructions, I assembled my samples, placed them in the EdvoCycler Junior, selected the right program, and hit run. The thermal cycler shifts from temperature to temperature, amplifying DNA. When the samples are done amplifying, I remove them from the thermal cycler and analyze them using agarose gel electrophoresis. The samples are loaded into the wells in our gel, and the combination of electricity and a porous gel matrix separates the DNA fragments by size. Since DNA is clear and colorless, I added the fluorescent DNA stain CyberSafe to the gel, which binds to the DNA and lets us visualize it with blue light. So, let's take a look at our results on the True Blue 2. What we can see is the only sample that amplified is the one that has everything in it. The others did not. Now, if we saw amplification in one of the lanes that were missing components, that could be a red flag. For instance, if we saw a product in the no template lane, it would suggest contamination in that sample, since Taq polymerase needs a DNA template to build new nucleic acid. So, through our experiment today, we showed that primers, template, and DNA polymerase, specifically Taq DNA polymerase, are necessary for the success of PCR. When we omit one of these components, the polymerase chain reaction cannot occur, and we do not amplify DNA. Thanks for listening. We pride ourselves on providing the highest quality customer service for our teachers. Call, email, or send us a message on social media.